Hi, I would like to thank the organizers for the opportunity to give a talk at this Evolution and Comparative Genetics conference. My name is Avital Sharir-Ivry from McGill University, and I'm going to talk about quantifying evolutionary importance of protein sites using an alternative measure that involves the structure of the protein. So within a protein, different residues evolve in different evolutionary rates. Some residues are more constrained and evolve more slowly, and we know that structural and functional properties have a very strong effect to create these different evolutionary rates. So as you can see in this figure, residues in the protein core evolve more slowly than residues on the surface of the protein, as the core is more constrained, and functional sites evolve more slowly than other sites as well. So overall, the general way we think about this is that if a protein site is important for the structure or function of the protein, it will be more conserved. It will be under stronger selective pressure. Recently, a remarkable strong signal was discovered, showing that there is a gradual linear increase of evolutionary rate with distance from catalytic sites in enzymes, and it has a strong long-range effect as the signal reaches up to about 30 angstrom away from the catalytic site and is affecting roughly 80% of the residues. So we then asked ourselves the following basic questions. Are these conservation gradients a general phenomena, and can they serve as an alternative measure of evolutionary importance of protein sites, alternative to classical conservation? Meaning, is it that the more a site is important to the structure or function of the protein, the stronger the conservation gradient it will induce? So in enzymes, we know that catalytic sites are much more constrained than other sites. Maybe it is simply that the strong selective pressure on the catalytic site propagates into the enzyme structure and creates these conservation gradients, and other sites which are less conserved will also create conservation gradients, however, much smaller. So to answer this question, we studied a large dataset of proteins of the yeast proteome. We calculated the conservation scores of each protein residue in the dataset and the conservation gradient that they induce in the protein structure, and we plotted them. So this plot clearly shows that there is a strong correlation between the two measures, between how conserved the protein site is and the strength of the conservation gradient it induces in the protein structure. Meaning, the more residue is conserved within the protein, the stronger is the conservation gradient it induces in the structure. And when we examine this correlation for each protein separately in the dataset, you can see the distribution of correlations, and it is mainly higher than 0.4 in absolute value. Since our dataset included a large variety of proteins from the yeast proteome, we can say that this correlation is a universal phenomenon and is not characteristic to a specific type of protein. So these two plots also tell us that selective pressure on a site generally propagates into the protein structure via residue-residue contacts. And they also tell us that conservation gradient indeed can serve as some measure for the evolutionary importance of a site. And when we look at different types of protein sites, such as buried sites and exposed sites, we see that this correlation can be seen within them as well, such that the more residue is conserved, the stronger is the conservation gradient it induces in the protein structure. And this applies also to all other types of functional sites, such as ligand-binding sites in enzymes, in non-enzymes, allosteric sites, protein-protein interaction sites, and catalytic sites. So indeed, this correlation between conservation and conservation gradient in the protein structure is universal, as it holds for different types of sites. In the plots that I showed you before, the x-axis were the conservation rank of the residue, which is a relative measure within the protein. We also calculated the average evolutionary rate of the corresponding sites. And you can see that the linear correlation between the evolutionary rate of a site and the conservation gradient it induces. Sites with low evolutionary rate induce stronger conservation gradients and vice versa. So now the question is, where are catalytic sites located? Remember, we started by seeing that catalytic sites induce very strong conservation gradients. So they're probably here in the upper left part because they have very low evolutionary rates and high conservation gradients. However, when we draw them on the plot, this is where they are located. The conservation gradients they induce are significantly stronger than the plot suggests. And what is going on with other types of sites? Buried and exposed sites are sitting on the plot. Non-catalytic ligand binding sites are very close to the plot. And protein-protein interaction sites and allosteric sites induce somewhat lower conservation gradients. But overall, the main point is that catalytic sites induce stronger conservation gradients than expected from the propagation mechanism of selective pressure, and that there is something else that causes catalytic sites to induce strong conservation gradients. In the previous plot, I showed the average gradients and evolutionary rate of different functional sites. Here we sampled randomly different subsets of these functional sites and measured the evolutionary rate and conservation gradients of each subset. So each point in this plot represents such a subset. And you can see that catalytic sites induce significantly stronger conservation gradients than other sites with similar evolutionary rates. Also, we examined all the proteins in the dataset that have both a catalytic as well as other non-catalytic functional residue. So in those cases where the non-catalytic residue is less conserved than the catalytic residue, for most of these cases, its conservation gradient is also lower, as expected from the propagation mechanism. However, when the non-catalytic residue is more conserved, only about half induce higher conservation gradients than the catalytic residue. So this further shows that catalytic sites do not completely conform to the propagation mechanism, and they have an additional constraint that controls the conservation gradients they induce. So to summarize, I showed you that there is a universal linear relationship between the evolutionary rate of a protein site and the conservation gradient it induces in the protein structure, and that this is a universal phenomena as it applies for all types of proteins and all types of functional sites. And these observations tell us that generally selective pressure propagates into the protein structure via residue-residue contacts, and that conservation gradients can serve as an alternative measure for the evolutionary importance of a protein site. However, I also showed you that the propagation mechanism cannot fully explain the long-range distance constraint from catalytic sites, and we believe that the origin for the strong gradient from enzymes is probably their unique function to stabilize the transition state of the catalyzed chemical reaction. And these results also point that if we combine this measure of conservation gradient with the classic conservation, it can provide us with more complete understanding of the factors that affect evolutionary patterns within proteins. And finally, I would like to thank my group at McGill and especially to Brandon. You can meet me at the poster session, and thank you very much for listening.